My guest today is Jamie Andrews.
Hi, Tom. Yeah, thank you very much for inviting me on. I've been an avid listener to your podcast for a while, and some of the topics of interest for me really is how I got started into what I'm going to be talking to you today about. Essentially, virology, what happened to us in 2020, and the project that I'm running, looking into some of the science and the laboratory methods that are behind what they call pathogenic viruses. The reason why they, locked 3.5 billion people into their houses and tried to force a medication onto people. In the country where I am, I moved to France. We had a vaccine passport for nearly a year's time, so I was excluded from society. My kids, wife were not allowed to go to restaurants, pubs, bars, even soft play centres. We were removed from society, as I think the story is for a lot of people. And so it was in my being, it was in my fibre to actually investigate the merits, the scientific merits on which that was built. And really, my journey into this segues very much into your channel and what you're doing and the types of interviews that you have, because funnily enough, my only real grounding in science, in authority, is in geology. I did a geology degree at Durham University in the UK, which is a pretty reputable institution. I didn't have much enthusiasm in the kind of higher education system, but I got through it. I got a degree, and that was my kind of entry level into it. Really, my interest peaked in what came along in the third semester, which was the only ability to actually look at some of the data ourselves. We were given anthropogenic climate data. We were given Vostok ice core data. It was, and I'm sure your listeners will be very familiar to this data, the most accurate paleoclimate data, which is oxygen 16, oxygen 18 isotope data taken from these ice cores in an Antarctic subterranean lake. And as an undergraduate, we were presented with this data and said, put it into a graphical format. It was something pretty loose like that. So we got given this data, and you map it, and it shows all the peaks and troughs of standard Milankovitch cycles, which is what you're taught about, that the Earth's orbit affects the precession over time of standard paleoclimate data, right? The only thing is that they gave us also, and they didn't tell us to map it, the CO2 data. I, as an interested kind of party, as an interested undergrad that, was curious, decided that I'll map it next to the oxygen isotope data. And would you know it, there was about a 600 to 800 year lag of identical peaks and troughs of CO2 data. And so I quizzically sat there as a 20-year-old saying, well, quite clearly, CO2 is not a driver of climate. And I remember distinctly telling my lecturer this kind of, have you done this? Have you plotted this on a graph yourself? And I remember it to the day, is that instead of actually saying anything, he tapped our notebook, which was sponsored by ExxonMobil. He tapped the logo, and that was it. And that's what I needed to know from then on in. The sponsors of our department was ExxonMobil. We had numerous people come in describing the Exxon slug, which is the way that they look for capped areas of oil, of crude oil. And so I knew then and there, really, it was the start of a journey. Because I left, I didn't have anything to do with, I was offered a master's, I was offered to go and do a few more bits and pieces into higher education, but I had no interest in it really. And After the few years, I started to dig deeper into what was the IPCC, the global organizations that were in charge of basically what I would consider to be covering up data that was contrary to media and contrary to political output that didn't match up. And so for the next nearly 20 years, it was very firmly rooted in my belief and my knowledge that the powers that be, shall we call them, rooted around the United Nations and the IPCC, which is the governing body for climate change within the UN, perfectly had it in their remit to, and I use this word, broadly, is commit a hoax that the Earth is going to either boil or freeze or do whatever they claim because of cow farts or because of carbon or whatever. I knew within my heart of hearts that there were the machinations of globalism that were capable of covering up this data. And so when it came to 2020, I had absolutely no idea about viruses, virology. I had no grounding in it whatsoever. It kind of piqued my interest when I found out that, I think we all found out fairly early on in February, WHO was driving the data that was behind the claimed pandemic, the WHO. And so my alarm bell started to ring. It was like, all right, okay, well, it's a UN government body. And I have visceral knowledge that these independent globalists, quote unquote, independent bodies that are all subsumed by the UN perfectly have it within their remit to start faking this data. And so I started to dig away first at the some of the data which was not particularly in hindsight was not when you look at epidemiological data. So when you have a death and they ascribe it to a virus, that's not actually assessing causality. That is assuming causality. So what they were doing, they were giving infection fatality rates, the IFR. For a few months, I had myself twisted up in knots as I had numerous pieces of data spread across the desk saying, well, the IFR over here in Bergamo, Italy, and the IFR just over the border in Austria was completely different, two almost identically geographical areas, only split by a political border, were showing these IFR rates, which were drastically different, which, again, to me, rang alarm bells. But really, it was the causality part of it which kind of irked me. And this started to really ring alarm bells when I had friends who were doing charitable work in India. So I had, in fact, two friends, both of them on the streets in New Delhi and Mumbai, respectively, working with very underprivileged people that were in the most conducive petri dish, shall we call it, for infectious diseases. They are there's malnutrition, there is disease, there is poverty, and in terrible areas for pollution, terrible areas for rubbish on the streets. And I got back reports from them. I was staying in touch with them through 2020, and it was very odd that they said that basically, given their baseline of what you would expect there to be a level of disease and illness, that did not raise one iota over the course of 2020. And so I kind of went, well, I had grounding in looking at the data, being able to assess peer-reviewed data, in my undergrad with geology. And so I went to Google Scholar, and I didn't really know what I was looking for in terms of the terminology. I'd done a biology A level, but apart from that, my grounding was okay. But in terms of laboratory work, in terms of exact nomenclature of what they were calling these things, I was typing in, putting viruses up people's noses on purpose and seeing what happened. So it took me a little bit of time to refine exactly what I thought I was looking for. And it turned out that it's called a controlled human infection model. And they did exist. I was a little bit surprised because there's a lot of claims of ethics, but very specifically in the starts of the 20th century, when maybe, because of the wars that were going on, ethics kind of got thrown out of the window, really. They did do a lot of experimentation where they took people that were infected. And this was early on, before they claimed to be able to culture a purified virus, what they considered to be a purified virus. They were taking what I would consider to be a quote unquote natural contagious pathway, which was taking people who were exhibiting the symptoms of what they considered to be, and one of the largest and most convincing to me early on was the Spanish flu. So it shared exact hallmarks with what was happening with COVID, in the fact of the political machinations, the forced masking, some of the isolation, some of the lockdowns were all employed during 1918, when they said that this Spanish flu killed, and some of the upwards estimates in the tens of millions of people. They were all done posthumously. Interestingly, the average age, they claimed, was 27, a male, and 27, which goes against every other respiratory disease for the rest of humanity, which was quite strange during the setting of a war in World War I, when the average age of fighting males is around, the mid-20s, and all of a sudden they're saying it's mid-20-year-olds that are only dying from the Spanish flu. They took these people as part of the Royal Navy sorry, not the Royal Navy, the Navy in the US, and under the direction of J.M. Rosenau, who was head of medics in the US Navy, they decided that they were going to try and show this was pathogenic, show that people would catch and die of this thing. And so they took 100 men, and they took people in the sanatorium who were in a real bad state, that they said from not from the gas poisoning, not from the very high levels of aspirin, not from the mandatory vaccines that they were pumping into the people, but supposedly from an infectious virus, where they were in bed suffering from these diseases, and they quote unquote gave these people infected material. So they took young, healthy men of fighting age, and they did everything that they could. They took sputum, they took mucus from these quote unquote infected people, and rubbed them in the eyes, the nose, the throat, the tonsils of healthy individuals. They got them to sit by their beds and have a direct discussion with them, very close up, to hug them and to get the infected to cough in their faces. And this was a real revelation to me. It was a jaw-dropping moment, in the fact that I read and read and read, and at the first end of 100 people, not a single person got sick. And so they made a few changes to the method. They said that, okay, when we were putting the infectious material up their noses, we were first sterilizing it with salt solution. So they took the salt solution out. They said maybe the salt solution is killing the virus. And at the time, they thought it was a bacteria. They said, okay, it's a bacteria. But really, all of these things were just direct from a sick person. So they were testing contagion in its fullest sense. And they changed the method. They redid the results in a different place with 100 new people, and not a single person got sick. And I'm sat there going, right now in 2020, we are locked down because they're saying that your neighbor can cough, or a person at the supermarket can stand too near to you within 6 feet and transfer this deadly disease. Yet when they're directly removing all of the ambiguity by taking infectious material and purposefully putting it up people's noses, nobody's getting sick. And so this was a real kind of life-changing moment for me, that all of a sudden I went from being very skeptical in the fact that, you know, maybe they were just fudging the data, maybe this was a mild virus, to seriously considering whether virology and contagious diseases worked in the way that they were claiming at all. And it didn't really stop there. And unfortunately, it wasn't just the one-off. I then went on to do what would be a literature review, trying to find every single human-controlled infection model since, which in the past 200 years, they've experimented on roughly 250,000 people. And the overwhelming majority, or in fact, every single one of them, does not get sick. And when I say that that's not just within influenza, that's not within flu, that's not within the common cold, that's within all of them. There are studies with some of the more scary ones: HIV, polio, measles, chickenpox. They've done them all, a lot of them through the early 20th century. So you have to really go back into some of the old manuals that are, transcribed from PDF and things like that. It's not so easy, but it was this moment of just throwing these things over my shoulder and going, but that one comes to nothing. That one comes to nothing. That one comes to nothing. Show me people getting sick from a virus on purpose. But I couldn't find it. And this is a fact. In the 250,000 people that have been experimented on, not a single person has been hospitalized. Not a single person has got bronchitis, pneumonia, a fever so high that they would be hospitalized, gone on to any sort of intubation. Now, later on, you will find that there is a turning point. 1954, which was a guy called John Enders, who developed a method to quote unquote isolate viruses. So he took a cell line that was cut out from a monkey and said that to propagate and to make this virus, you can't just pull it out of somebody's nose and you have virus. You can't just spin it in a centrifuge. There's not enough of it, they claim. They claim that they have to grow it, which is fair enough. You can see bacteria, you can see these things under a light microscope, and they need to feed like scavengers or other parasites. They need to feed on things to be able to grow. And so the logic was there in the fact that these things that they can't see yet because they didn't have the microscope power to see them. It was a complete assumption, by the way, that viruses existed. They just said there must be something contagious in this sample. So John Enders came up with this protocol that was actually supposedly an immunological paper to start off with, and really an afterthought. But he took this cell line and he bathed it in infected material from a patient that he said was suffering with measles. So they had the symptomology of rashes and a high fever. And he put this in the cell line, and the cell line died. Okay. And he noted that the cell line died. And so he said, that must be because of viruses in it killing the cells. The problem is that in his very own paper, when he designed this isolation and purification method of a virus, he also had a control. So anybody that's a scientist or anybody that should be interested in science should be interested in a control, because the control is really what shows you is science, is happening, is blamed on the independent variable, i.e. the thing that you are studying. So he conducted a control where you remove the agent that you're studying to show that it is the independent variable or the agent that is causing the death. So he had an uninfected control. He had the cells, he had some of the nutrients that they put in to grow the cells, but he didn't put the infected thing in. And in his own paper, it says, we still observed the cells dying. So hopefully the logical of mind should say, well, clearly it's not what's in the sample that's killing the cells then. Maybe it's the environment that you're keeping the cells in that is not conducive for their growing or even living. But instead of doing this, he said, well, I can see a difference between the dying over here and the dying over here, so we've isolated a virus. And this was gold stamped into virology. 1954, John Enders came up with the gold standard protocol for viral isolation, and it has been used ever since to isolate a virus. Fact. Now, the grounding has been built on this idea that from ever since that they have in a vial a purified virus, a thing, a causative agent. And some of the earlier, as we were discussing, the earlier Contagion studies where they were taking, and in my opinion, it's the best natural pathways where you're trying to discover where people are actual contagion. Can you sit next to somebody and can you infect them with your cough or your sweat or your breath? These were the closest to natural transmission pathways. Whereas in 1954, there was this sharp change towards the fact that they said that they had the isolated, purified thing. And so they started to do these controlled human infection models where they were taking this vial of stuff and then putting it up people's noses. And so they were claiming that they had purified pathogen. And unfortunately for them, they kept failing. There was even during this time, slightly before and slightly after, there was a common cold unit in the UK, in Porton Down in the West Country. And they saw some 40 or 50,000 people over the course of a few decades come in where they were studying the cause for the common cold. And there's a wonderful BBC video where they actually interview some of the people that come to stay, because there was a bit of a government scheme. These people got given money to come and be experimented on, and they interview a few of the ladies, the ladies of leisure that go down there. And in typical kind of 1950s and 1960s British broadcasting tone, he says, hello, how are you doing, Doris? And Doris goes, oh, I'm fine. It's lovely. It's really nice here. I get to walk in the countryside. I'm actually coming back here in a few weeks' time because it's such a nice time with my friends. It's like, hold on a second. Aren't you meant to be experimented on with deadly viruses? But no. These people actually came to this, what they saw as a kind of holiday camp, because in the own words of Nigel Dimmock, the head virologist that was there for nearly 20 years, he said the chances of you actually getting sick are really remote. And so they were putting what they considered to be purified virus, not just the sputum, the fluids of sick people. They were taking the spun down the exact etological pathogen directly up people's noses. And they did that right the way through to this current day, and unfortunately have failed. And this was, again, a kind of a problem that arose to me is that I was gathering this information through 2020, and at the time plastering it everywhere on Twitter. And at the time, I got myself into a little bit of trouble because I found a very modern one where they studied influenza A, which was conducted by Jonathan Van Tam. And Jonathan Van Tam at the time was the chief medical officer of the UK during the pandemic, the scamdemic. And he had conducted this influenza A trial, and not only had nobody gotten sick, nobody had even tested PCR positive when they had put what they considered to be purified virus up people's noses. And even the title, and within a lot of the titles, they managed to kind of obscure these negative results. It's always, we could learn more from this. We don't really know why it could be this or that. It's never this is a fail, right? But in this, there was really no way of getting out of it. It was conducted in 2019, I think it was, and they released the results around the time of 2020 happening. And so they had to kind of give it a muted response, and it was entitled Minimal Transmission. Now, it wasn't minimal, it was nothing. There was no transmission whatsoever. And I was plastering this everywhere over Twitter to anybody that would listen. Why are we locked down when they have no proof? Why are we locked down when they can't show that there is transmission or contagion of biological pathogens? And my account was nuked. I was taken out. I had 25,000 followers at that time, and it was a kind of deletion from, and I'm sure that a lot of your listeners will be in the same kind of boat when it comes to screaming, from the rooftops about the anthropogenic climate change or climate crisis, revealing of the fact, that not is all as it seems. I think people will be aware that that is very much happening within not only the realms of professional science, where people get hounded out of their professions, but also to the layman just discovering, because I was in a fortunate position that I had no skin in the game when it came to virology whatsoever. It was one of those things that I just approached as a layman. And so being able to observe it with no preconceived notion and no kind of end result of, this must be an assumption, like viruses must exist, therefore there must be contagion, so on and so forth. I had the luxury of being able to view these things with an open mind, with a blank canvas, as it were. So it was a bit of a kick in the teeth to lose, a following of that size, but it really gave me wind in my sail to pursue this, as saying, well, if they're going to silence my voice, they must have a reason why. They must have a reason why they don't want this information to be out there. And at the time, I was part of a few Telegram groups with a few friends that were investigating the fraud of virology at the same time. And part of that, we were actually starting to get some interesting people joining us, because they were people that were within the industry. They were virologists, microbiologists, biochemists. They were coming forward in private, kind of saying, look, I'm in this industry, but I'm actually I'm interested by what you're saying. And the more and more I look at it, the more I can't make head nor tail of this. And so a group of us started to look into actually the laboratory process of viruses. Like we said, in 1954 with John Enders, the whole thing seemed to have a pretty fragile grounding on the actual laboratory processes to make, quote unquote, make a virus. And so we started to look, and we found a few people historically. And at the time, there was a guy called Dr. Stefan Lanka, who came into notoriety to our interest at about that time, because he was conducting what he considered to be control experiments. And we all knew, as looking at this groundwork with John Enders, what a control experiment was. And Stefan Lanka was doing this on designing an experiment that had all of the hallmarks of trying to falsify the laboratory protocol in virology. And it was interesting because he was a virologist himself. He had a seasoned career, 20 years in industry, looking at bacteriophages, which are a claimed virus that's specific to bacteria. And so he had that long burgeoning career, yet had come to the conclusion after many years that, in fact, viruses didn't exist. That was his conclusion, and that's his conclusion now, is that not only is, contagion not really set in stone, and in fact is not reproducible in any fashion, but maybe the actual causative agent didn't exist at all. And again, this was kind of another kind of layer upon the journey into this that I wasn't really expecting. I hadn't really thought about it before. But Stefan Lanka, when we started to find out a little bit more about his story, had been right to the top of German court, the German Supreme Court in 2016. He had offered a wager to try and raise awareness about the dangers of the MMR vaccine in Germany, where he was a national. And he offered €100,000 wager for anybody to present evidence of the measles virus existing. And people might go, "What do you mean?" I'll take that. It's €100,000, just to show you that something exists. It's sure, there you go. And somebody did. Somebody came forward. It was a lecturer called Dr. David Bardens, and he presented 6 papers and said, "Hand us over the cash. Show me the money." And Stefan refused to pay. And so it went to court. And there was a lower court of Ravensburg, and they put across a statement which said that the measles virus was present in these things. They brought in an independent microbiologist to act as a scientific expert in the trial, whom initially sided with Bardens, but he made a few clinical errors. He said that in no one paper there was evidence of the virus. When you look in all of these papers split across them, then potentially there is, but in no one paper there is the size, the genomic sequence, the shape and pathogenicity displayed in one paper. So he said the Koch's postulates, which are a very basic level of proving the existence of a pathogen. Can you culture it? Can you purify it? Can you put it up somebody's nose? Can they get sick? And can you then reharvest the pathogen? These are, very basic criteria for proving a pathogen. It's used for describing a new pathogen, but it should be, just a description of any sort of biological entity, really, when it comes to pathogenicity. And he said, and he made a bit of an oopsie, because he said that no, it doesn't fulfill Koch's postulates in any one paper. And so Stefan Lanker's original original wager was, can you provide me evidence of any evidence in a single scientific publication of the measles virus existing? And so he lost the first court case in the lower court of Ravensburg, and he appealed and went to the higher court of Stuttgart. And the higher court of Stuttgart said, essentially, on a technicality, because you've asked for one paper, we have to say that the prize money is not to be awarded because you have not proved the existence of the measles virus in a single scientific publication. Now, the media will say, and Reuters did a very huge cover-up on this. He won by technicality. This was wordplay. It was a single paper. Viruses do exist, but it was done through nefarious means, which is half right. It's half right because the criteria was strict, that it was to be in one paper, and there is not a virological paper that demonstrates this in a single paper. Now, there's kind of two questions that come from that is that why can they not demonstrate that something exists in a single paper? I can demonstrate to you that an orange exists in a single paper, in a single photograph. It exists. There you go. Why should you need such rigorous, such lengthy data to prove that merely something exists? It's quite an oxymoron, really. If it's fairly evidential that it exists, then you should go, there it is, there's the proof. So there should really be no reason to be able to demonstrate it in a single scientific publication, yet there is no such thing that exists. And secondly, what they did by admitting that there was no evidence within a single scientific publication was that they admitted that the whole genome sequence, therefore the PCR test, was not proof that the virus existed, because one of the papers was the whole genome sequence. So they said, inadvertently, this German court said, the genome sequence of the measles virus does not constitute it existing in and of itself, which is quite a big one when in 2020 we were being told that these PCR tests were being the sole reason for the cases, for the deaths, because you were getting in hospital protocols. People were coming in having fallen off a ladder or in a road traffic accident, or dying from cancer, or dying from old age, or some of the horrendous protocols that they were giving people of drugs in the US, remdesivir and intubation, and in the UK, midazolam and morphine. And it was a killing field in some of these old age homes. And then they were coming along with the PCR test and going, 'twas the virus that killed them. Well, was it? Or was it the pipes that we were shoving down their throats and the kidney toxic medication that was causing them to die, And so this PCR test was really used to, what I would say, fabricate the idea that there was a spreading, transmissible, deadly pathogen. And so myself with a few friends, should we actually investigate this ourselves? Because Stefan Lanker, off the back of this court appearance, did a control experiment. So he did essentially what John Enders did, which was take some of the protocols that were used to quote unquote isolate a virus and not put any virus in and see what happened. And we had a few people that were in industry. They knew how contract research organizations operate. And so we kind of said, okay, well, there's no, limit. We don't have to be trained virologists to pay independent research labs to look at this stuff. They're pay for play. Contract research organizations around the world will happily do the science for you, happily look at things. That's what they do, right? And so again, this was another kind of life-changing thing for me is the realization that maybe actually we could do some sort of, project to actually look at this for ourselves, because all of the data there, when you're relying on published peer-reviewed data, there is a real, how would you describe it? I don't know, cultish mentality behind the peer-reviewed publishing system, because they don't really like dissenting opinion. They will very rarely even acknowledge dissenting opinion, let alone publish it. And so we wanted to find out for ourselves. And so we developed these protocols, and we crowdsourced. We went out amongst our group of, at the time it was kind of growing. There was about 50 or 100 of us maybe in this group. And we had a bit of a whip around, and we went, okay, if we all give in $100, we can do this. And that's what we did. We engaged the CRO. We gave them the protocol. They said, the protocol's fine. We did a lot of research into it to find out the standard laboratory methods and techniques. They had little to no questions, about what we were looking for. Essentially, we were kind of blinding them a little bit to make sure that it wasn't politicized. We said that we were looking at extracellular vesicles, what happened when you starved a cell line. And so what we did is we went into and this comes all the way back to the UN, is that the UN and the Clinical Laboratory Standards Institute, which writes the M4A1 manual on laboratory protocols. So there's a top-down governance of how virologists isolate a virus, and there's pretty strict guidelines as to what they're doing within these cell cultures. And we followed those protocols and gave them exactly to an independent lab. And they came back, and what we found is that out of the 90 cultures that we did them in, so we had 90 cultures of a human embryonic kidney cell, which is used to isolate, quote unquote, adenovirus SV40. They broke down. They had the exact cytopathic effect, CPE, which is a noted morphology. So when the cells grow, they grow out in a certain way. They look a certain manner, rounded. They look coherent. Yet when they die, supposedly when a virus is put in, they die in a very specific shape and a very specific pattern. So you see things like rounding, which is where, as it sounds, the edges of the cells round off. You see plaquing and cincture, which is where the cells merge together and raft together. You see cytoplasmic inclusion and granulation, which is where the cytoplasm breaks down and you get a kind of blotted effect within it. You get ballooning, which is where the cells actually lift off, and you get apoptosis, which is where the cell dies. And so these are all noted in this UN regulated manual as being specific to viruses that they put in there. So they can say, okay, SARS-CoV-2 causes rounding. That is one of the things. If you see rounding of cells, you have SARS-CoV-2 in your dish. If you see cincture or plaquing, you have this type of virus in your dish. And we noted every single one of those CPE morphologies in the dish without putting a virus in there. And this is the important part, without the possibility of a virus being in there. Because when you buy a cell line, when you buy the reagents, when you buy all of the ingredients that go into this cell culture, you are buying a guarantee. It's very expensive stuff to buy just a tiny little slither of a cell line. Unfortunately, they come from really horrible places. This one comes from a fetus, a human embryonic kidney. But they guarantee you that they're sterile. So when they cut them out, they PCR test them and they say, what you have is a clinical blank canvas for you to for you to work from. When laboratories are in clinical studies, they have to know that they have a blank canvas and that the independent variable of whatever they're doing, they're testing a drug, for instance, that it's the drug that's doing the work, not a virus, or vice versa. And so we know when we bought this within a chain of custody that they deliver to you in minus 80-degree frozen state a completely blank canvas. And so there is no possibility of any pathogen being within this. Yet we saw unanimously in 90 cell cultures it breaking down to in the exact timing with the exact morphology of a virus being present in there. And so we started to look further. We went back to a CRO and we conducted some microscopy work. So viruses can't be seen with a light microscope. They're too small. They're on the nanoscale. You have to effectively kill the cell line. And here's another bit of conflicting logic that goes on. When you hear from virologists or hear the story around how a virus operates, they say a virus is an intracellular parasite that moves into a cell, replicates, and then comes out. Well, let me tell you, it's impossible for them to know that. And the reason why it's impossible for them to know that is the fact that the nanoscale of a virus is impossible to see in real time. So with a light microscope, you can see a bacteria moving around. You can see it being motile. You can see everything to do with it being living and how it replicates. With a virus, it's too small. You have to go into transmission electron microscopy or cryomicroscopy, where you kill and fix a sample. So there is no possible way that they could ever see with their own eyes a virus moving into, replicating, and then coming out. Even if you wanted to try and conduct very quick slices, so you take a slice, you fix it, you put it in, you will still always be seeing a fixed, motionless picture. So the idea that they say that they know that a virus can move into a cell, replicate, and move out is quite clearly nonsense. So we went to a microscope lab, we took those uninfected cultures and we gave them, and we said, can you image these cultures? They gave us back 6 images. And I mean, at the time, we've gone on to replicate this experiment again. But at the time, we had limited budget, limited funds, but we wanted to see the visceral particle that they call viruses, or indeed, we shouldn't have seen a particle that they call viruses because there was no possibility of a virus being in there. They gave us back these 6 images. We gave them the blinded protocol to, quote unquote, look for extracellular vesicles. So just a round blob that comes off of the lipid membrane of a cell. When it breaks down, that lipid comes out and it's slightly hydrophobic, like a lava lamp, like an immiscible oil in a water. It forms round type objects with a liquid around it. And so an extracellular vesicle is just a non-filled vesicle, a vesicle that is essentially an air bubble. And we said, can you find some of these? They took a few pictures of those, but in the corners of these 6 pictures, I was looking at them. What's that? Well, that looks exactly like SARS-CoV-2. And that looks exactly like HIV. And that one looks exactly like measles. We found 3 supposedly pandemic, epidemic causing viruses to the exact shape, to the exact size, to the exact protein inclusion, to the exact protein coating. The same morphology. You get that round with the spiky bits around the outside with the SARS-CoV-2. There was a particle in these 6 pictures that looked identical. There was it's 100 nanometers in width. So we got out the ruler, we showed that given the bar chart, it was exactly the same size, exactly the same shape, And so we took this further. We repeated the entire experiment. We took it to a completely independent lab once again. In fact, the first session we got the independent laboratory to video the whole protocol to know that it happened, to know that it was, because there's obviously pretty skeptical people online that kind of go, well, they just call our bluff on anything really. So we went the extra mile to say, look, this is it happening. You can say that there's bias, you can say that there's starting out with a biased opinion, but this is the scientist that conducted it in his hands. These are the methodological processes that he followed. Please show us where we're wrong. And so we did this again, and we went further, this time using the exact protocols of SARS-CoV-2 isolation, which is a monkey kidney cell. Ironically, they couldn't get SARS-CoV-2 to break down in a human cell. They had to use a monkey cell, which again should really ring alarm bells for any of those with logic out there, that if you can't make it kill a human cell in a Petri dish, how can you get it to kill a human in real life? They could only get it to kill or supposedly break down the more fragile Vero cell, an African green monkey kidney. And we conducted the identical protocol for the very first isolation of quote unquote SARS-CoV-2, came out with the exact same results, unanimous CPE morphology seen in the uninfected cell lines. We then took them into a 2-hour session. So myself and Albert Matthews, who conducted the experiments this time, we worked with the microscopist, and live we got to move around in transmission electron microscopy, instructed where the microscopist should zoom in to be able to find 27 different viral morphologies present in the uninfected cultures. Everything from Ebola to monkeypox to smallpox to HIV. These things look identical. And we then took these results, we put them out, we've put them all open source. They're all open source and crowdsourced for people to download, to look at, to repurpose into their own ideas and opinions on the results that we've made. Me personally, I did a write-up. I then got AI to operate as an objective mainstream viewpoint, because unfortunately I can't get any rebuttal from mainstream virologists. They refuse to look at it. We have been desk rejected on the 2 or 3 journals that we've submitted this to on purely political grounds. That's it's hilarious really that the majority of the reviewers that desk rejected it because viruses exist. Literally, that was the reason why they gave it. Nothing to do with the protocols, nothing to do with the methodology, nothing to do with actually analyzing any of our results, just purely the fact that they believe viruses exist, therefore they will desk reject it. And so we can't really get any sort of feedback from the mainstream. And so we rely on crowdsourcing, we rely on AI objective verification, and it did just that. When I trained ChatGPT on the exact physical morphology that was seen within a Petri dish, it confirmed that we found identical morphology, identical shape, identical inclusions, identical coatings of 27 different pandemic and epidemic causing viruses. And so my conclusion is that when you falsify something in the physical realm, when there is a physical particle that they are claiming is a virus, yet you find that identical physical particle, you must conclude that it therefore does not exist, because they are mislabeling something. And what they are mislabeling is standard cellular debris. When the cell line breaks down, all of the interior spills out into the surrounding fluid, and it's like a Rorschach graph. It's like a Rorschach ink blot test, where they point at these things and go, that looks like a SARS-CoV-2, that looks like an elephant, that looks like a whatever. They are looking at clouds, they're looking at this patterned idea and ascribing these scary stories to what is standard cellular debris. And that really kind of brings me up to where I am within the project. Now, I've done genetic sequencing to further that, because the downstream methods are really ended up they concertina, and they pinpoint into the genetics realm. And so this is the part which is the where it all comes together. And so I'm working my way through that in private as we speak. It's some pretty big stuff, but we're going to be releasing that this year to go through with some similar astounding results. So that's really where the project is going. That's what I've been running for the past 2 years, looking into this subject. And as we discussed before, if this piques interest, I'm more than happy to come back with some of that data to actually walk people through it in kind of finer detail. I know that the topic is, it may be new to people, and it may be new to some of your listeners, and especially some of the laboratory protocols that need to be kind of explained a little bit more in depth. But I'll be happy to do that if you were interested.
So can you get anybody at all to explain what these 27 particles are doing there in samples that should not have them? They just will not respond. No, it's a cognitive dissonance that I think that they know that if they do respond, then we're debating and we're talking, and they know the logical conclusion. The rebuttal from virologists and from AI is that, yes, and there's plenty of peer-reviewed papers out there. There's one that sticks out in my mind, which is COVID-like particles found in negative renal biopsies, which was a 2019 paper where they wholesale admitted that, yes, there are these identical particles that are found in places which cannot possibly contain them. So they PCR tested people back in 2019. They came up negative, yet they were finding these particles. And so when it comes down to EM morphology, they say, well, we don't know until we PCR. So they take it completely away from the microscopy, from the physical realm, and take it into tests. Now, my rebuttal to that is that a test is an inference. A test is still not direct evidence of something existing. It is only an ascribed inference to a test. So when the PCR is conducted, they put in a fluorescent dye, and when that fluorescent dye fluoresces, they say, that's because there's genetics in there that we were looking for. Well, if you're to be really bloody minded about this, the fluorescent dye has just fluoresced. You could ascribe any story to that because you cannot see on a molecular level what's going on. And this, in my opinion, is what biochemists are doing when it comes down to all of these, is a very elaborate story, a very convincing story to some, that the tests are exactly indicative of what they're describing. And the results that I have, that I will be releasing this year when it comes to specifically genetic sequencing and the PCR test, is that's not correct. I have proof, and I can't go into it in too much detail because I'm legally bound not to at the moment. But we have proof that the PCR test is not measuring what it claims to measure, which is distinct sequences of nucleotides. So if I could leave you with that kind of cliffhanger, I know that there are plenty of people that have been with me for the 2 years that are desperately bored by the fact that I can't quite reveal it yet. But it will come to light. I'm just kind of dotting all the I's and crossing all the T's to make sure that really when it comes to this project, it is the final piece of the puzzle. And so, yeah, that's really where we are with the genetics part. But to answer your question, they move it into the realms of genetics and say, yeah, there are particles that look identical, but it's all down to the PCR. So they have put all of their eggs into the PCR basket, which is why I think that it's so important that this genetics part is done in a watertight fashion.
So I just wanted to throw in one thing from my own experience that reminds me of what you're looking at here. It's about the ivory-billed woodpecker. There was a peer-reviewed paper maybe 20 years ago, and they had a 6-pixel ivory bill. They had a picture out in the woods with 2 blacks, 2 whites, and 2 blacks, and that was supposed to be an ivory bill. But then people were pointing out you can take a picture in your office, and you can find ivory bills all over in your office with that same pattern. So it's kind of a similar thing.
That's it.
Yeah.
That's it. I mean, when you look at, you know, and I'm investigating DNA as well, which is, you know, a whole nother story. When you look at the Rosalind Franklin B51 picture, it is just that, you know, it's a group of students managed to recreate that using the spring of a ballpoint pen. You know, so it's a dangerous game when you're playing with inferences, when you are not showing direct evidence. And I think that in terms of being objectively scientific, I think that as a populace is where we need to be, in the fact that if you are going to say that 3.5 billion people are to be locked in their houses, you better have direct evidence of this stuff existing, direct evidence of it definitely causing disease, you know, because they put SARS-CoV-2 up people's noses. They did a controlled human infection model. It was published in Nature in 2023, ironically by Jonathan Van Tam, and they had to admit the title of it is in Nature magazine. We tried to give people COVID and failed. They gave 56 people what they considered to be purified SARS-CoV-2 up their noses, and nobody got sick. And so we've got to be more, you know, skeptical of this thing that they call settled science, because just, you know, from my cursory searches as a layman, it was anything but.
So you have time for one more question. I have lots
course,
more,
yeah.
but let's make this the last one. Just what do you think really did happen with the case of like smallpox and the Indians in North America? What did happen there?
Smallpox, I mean, you know, this is getting into the realms of what really makes you ill, right? So, and it's a question that I get nearly daily, and it's something that I wrestled with for a little bit of time, but I came to the conclusion that it's very, very simple, that what really makes you ill is what all of the mainstream tells you makes you ill. It's poor diet, it's drinking, it's smoking, it's chemicals in your environment, it's what they're spraying in the air, in the water, what they pharmaceuticals, drugs, all of these things the mainstream tells you makes you ill. It's just not a virus. It's cutting out that middleman. Once you accept that the environment, the terrain that you're in, the things that you're eating, the psychological stresses that you're under are causing those natural detox processes. And when you look at what actually is the flu and all of these symptomologies that you get are a detox pathway. When something goes in that your body doesn't want, a toxin, a chemical, it must come out, and it comes out one of a few ways. You excrete it out through defecation, urination. And if you can't do that, if it's assimilated into your bloodstream by kidneys or liver, it's in your bloodstream, and it can't come out via those mechanical ways. It has to come out via fever. So your body heats up, it opens the pores in the largest organ of your body, and you sweat it out, or you cough it out, you expel it through the lungs. It's put into the mucosal pathways where you can get decent transfer into the mucus, and you cough it out. And so the flu is a detox mechanism. Every time your body comes to a time when it is prepared. And this is one of the main things. This is that if you notice, the flu comes when almost you relax. One of the things that I point out epidemiologically is teachers will go through an entire term time being highly stressed, working every day. And then they get to the holidays and they get the teacher's flu. As soon as they stop doing the work, as soon as their body has time to detox, that's when it starts coming out. And so when you look at things like smallpox, and I mean, smallpox is quite difficult because circumstantially the evidence is quite difficult to see. When it's neurological damage, for instance, with polio, there's DDT, which was an organophosphate, which causes neurological symptoms. They were spraying it all over the crops and all over agriculture. And so you can marry those two things up very, very easily. They're plotted on a graph that directly correlate between DDT usage and symptomology of polio. Smallpox, when you come to it, because it's defined in an arbitrary manner, because any kind of rash on your body, any kind of lump or bump can be described as anything from the orthopox viral family, which is 20 or 30 different ones: monkeypox, orthopox, smallpox, and so on and so forth. And so you get a blemish on the skin, and they could call it whatever they want. And so usually when these things are occurring in distinct clusters at distinct times, it's from distinct chemical poisonings. So during that time, a lot of people used lead and arsenate. Again, with things like, and it's not a virus, but anthrax, they called it wool sorters disease because only people would get and die from anthrax who worked in sheep processing facilities, wool processing facilities. And what do they use on sheep? Well, they used arsenic dip dye. And so it's my belief that all of these small clusters of aggressive symptoms come from industrial poisonings. So uses of heavy metals in industry, in agriculture, and within household products where they're rolled out nationwide or countrywide or worldwide. Things like asbestos, when they take them out, they go, oh whoopsie, it causes lung carcinoma. And I think what they're doing is that they're using the germ theory, the model of germ theory, to cover up these industrial harms, to use them as a scapegoat to say, well, it wasn't me, Gov, it was the virus that undid it. So with smallpox, with the case of the sharing of the bed sheets when it comes to that, I think is just a tall tale. When they've conducted any other control experiments, and Hess and Unger did it with measles and chickenpox, where they actually lanced the boils of chickenpox, and then, I mean, it's gross, but they lanced them and spread the actual pus into healthy children and got nothing. And so, when you are directly studying those things when it comes to smallpox, just off the top of my head as well, there was a doctor called Dr. Rodemond, who again conducted a pretty gross control experiment of his own. He was dealing with smallpox patients, and he took the lanced pus of smallpox patients and smeared them on his face. He did this for some years, putting bits of this supposedly infected smallpox material in his beard, and went and treated other patients. And he said in his own notebook, in his own writings, that he saw nobody getting sick from smallpox, the disease not transferring from person to person. So sometimes it does take the pretty gross gross things to find these things out. And I have some friends that have got some pretty horrible party tricks of putting swabs of things in their mouth and stuff to demonstrate that Contagion is a hoax. But sometimes, you've got to do these things to be able to do it. And really, if you go with this in mind, and this is what changed my mind, is my own control studies where my children or my wife or myself were getting sick, and you really go and look with open eyes and actually look to see who is getting sick, when and where. And it's never 100%. It's never everybody at the same time. It's never everybody. And so, the immune system is another one that we can get onto, but it's used as the scapegoat to kind of get out of that. So that's a very long-winded answer to the question, but yeah.
All right, let's go ahead and wrap up there. In the show description, I put a link to your X handle, or your X handles there, and your Substack, your Control Studies Substack, and people can follow up your work there.
Brilliant. Thank you.